Okay, let's take a look at question number 3a. Describe the steps involved in acute inflammatory response. Okay, what are the 3 steps involved in acute inflammatory response? Well, we have activation of resident cells, vasodilation and permeabilization, recruitment of neutrophils and monocytes. So let's move to the start where we get infected by a pathogen. So if a pathogen was to breach our tissue through broken skin or things like that, they will first encounter macrophages and dendritic cells which have pattern recognition receptors capable of recognizing pathogen-associated molecular patterns of each and every pathogen. Once recognized, resident cells, mainly dendritic cells and macrophages, will release substances like chemokines, cytokines, lipids, and proteins. Okay, each of these are involved in subsequent steps. For example, cytokines TNF-alpha and interleukin-1 are responsible for increased permeability, activation of paracrine cells, and increased extravasation. Chemokines like interleukin-8 is going to be responsible for your third step, which is recruitment via chemoattraction. Lipids like prostaglandins and leukotrienes are going to lead to vasodilation and increased permeabilization respectively. Proteins like protein C5a, which is an anaphylatoxin, and histamine is going to lead to increased permeability of the blood vessel wall. So why is the second step so important? Well, vasodilation means an increase in blood vessel diameter. An increase in blood vessel diameter will allow more blood to be carried to the site of infection, hence more immune cells to be carried to the site of infection. An increase in permeability is also important because we want to increase extravasation of immune cells from the circulation into the infected tissue. As for recruitment is going to be caused by chemoattractants like interleukin-8 mainly, which can be detected by neutrophils and monocytes, causing them to perform diapedesis to go from the circulation into the infected tissue. And this is done mainly by 2 receptors, LFA1 and ICAM. ICAM will be found on the endothelial cell of blood vessel wall, and LFA1s will be found on neutrophils and monocytes. LFA1 mainly lymphocyte function associating antigen 1, and ICAM being intracellular cell adhesion molecule. For the next question, how does chronic inflammation lead to joint destruction? Okay, where was I? Okay, yeah, how does chronic inflammation lead to joint destruction or rheumatoid arthritis? Okay, well, we know that our acute inflammatory response needs to be shut down, or it can lead to dire consequences like rheumatoid arthritis. So what is rheumatoid arthritis first and foremost? Well, it's characterized by 4 of these: a thickened synovium, inflammation, angiogenesis, bone degradation, and cartilage degradation, each of which I will be going through on a cellular level, sorry. So how do we get thickened synovium? Well, monocytes or macrophages are going to release tissue necrosis factor alpha. Tissue necrosis factor alpha is going to lead to an increase of synovial-like fibroblast. Synovial-like fibroblast leads to thickened synovium. Thickened synovium or synovial-like fibroblast proliferation also leads to an increase of rank ligand expression. Rank ligand then binds to rank receptors on osteoclasts, and osteoclasts are responsible for bone degradation. Okay, and synovial-like fibroblast proliferation also leads to an increase in proteases, which are responsible for cartilage degeneration. Tissue necrosis factor alpha also leads to an increase in inflammation and angiogenesis, which is the development of new blood vessels. We not only have tissue necrosis factor alpha, as we also have rheumatoid factor, which is a type of antibody released by B cells. And this rheumatoid factor will form immune complexes around the site of joint destruction, leading to recruitment of more immune cells. And you also have T cells that release cytokines and chemokines, which attract even more immune cells to the site of joint destruction. Next, for the disease modifying antirheumatic drugs. Okay, the disease modifying antirheumatic drugs I recommend you guys to go with is methotrexate. Why methotrexate? Well, methotrexate is an antifolate, and to understand the mechanism of methotrexate, we need to understand the folate pathway. Okay, as we can see here, dihydrofolate reductase catalyzes the formation of dihydrofolate from folate. So folate forms dihydrofolate via dihydrofolate reductase, which forms tetrahydrofolate via dihydrofolate reductase, which forms methylene tetrahydrofolate, which can go two ways, either to go with purine and pyrimidine synthesis or reform dihydrofolate reductase via the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate. Methotrexate intervenes with dihydrofolate reductase, preventing all of these steps and preventing purine and pyrimidine synthesis, which is very important for DNA synthesis. And DNA synthesis is even more important for mitotically active cells like B cells and T cells, hence down regulating their proliferation, hence creating an immunosuppressive effect. Methotrexate also inhibits ICAR transformylase, ICAR being amino imidocarboxamide nucleotide transformylase, leading to an increase in ICAR, leading to an increase in adenosine. Adenosine will then bind to adenosine receptors, leading to an anti-inflammatory response. So methotrexate cancels all of these out. You're only going to have that reaction right there. As for biologics, the biologic I'm going to go with is anti-TNF. Why anti-TNF? Well, I feel TNF plays a major role in rheumatoid arthritis. So anti-TNFs like adalimumab and infliximab bind directly to the TNF, preventing its ability to bind to TNF receptors and bring about a response. Hence, those are the biologics and disease modifying antirheumatic drugs.